thanks everyone for attending today's session. Today we have a very special guest from Germany. So they run a program called LDC and they will talk about that. So this program helps academics, or it works with academics in advancing small molecule therapeutics. So over to you, Matthias.
Okay, so thanks a lot for everybody for joining and your interest. So just a few words for my background. So I'm a scientist by background, actually. I'm co-educated in finance and administration and co-founded companies in the past. Now I have 4 companies that I have founded over my whole career. I was also 16 years actually in technology transfer, working for the Max Planck Society. And one of my jobs was to create an incubator for projects within the Max Planck Society with a therapeutic underlying. And what you hear today is the result of this. The whole venture started 16 years ago. And then we have created the Lead Discovery Center, which started actually as an exclusive incubator for the Max Planck Society, but today is absolutely open. Just trying to switch to the presentation mode. Yes, it's absolutely open, let's say, for collaborations with almost everybody. Can you see my screen in presentation mode?
Yes, no. It didn't get bigger, but we can see your screen. I see that you hit the button though. That's weird.
Okay. Sorry, I'm just have... I cannot activate my laser pointer. Let's see. Okay, so actually the table of contents for today's discussion is really, I will introduce the LDC, our business model, the indications we're working on, tell you something about our portfolio and some success stories. But regarding the business model, really, I'm also focusing, let's say, about potential, let's say, the decision to do a straight license with existing companies or create a new company. I will discuss about the pros and cons and the stages that are ideal for these kind of business models. And also have a few slides regarding the infrastructure that we are offering for collaborations, and we'll, let's say, also introduce the different disciplines that are necessary for a proper drug discovery approach. And I will tell you a bit something about some pitfalls, because you can do a lot of things really wrong. So this is also my personal experience. I know when I started at Max Planck, actually, all the researchers that had these interesting projects, they did their own drug discovery, so to say, with CROs and so on. But at the end of the day, most of them really failed because they didn't do it really properly and professionally. And I think the most important thing is really to integrate all these different disciplines, to have really a kind of a project management team in place that avoids all these pitfalls, because you're sinking, of course, a lot of really capital when you do this really wrong. And also, let's say, we'll talk about project proposals that we are looking at, and of course, requirements also to collaborate with us. And anytime when you have a question, just try to give me a hint via the chat maybe, or just jump into it and you can ask anything at any time. So the Lead Discovery Center, actually, as I mentioned already at the beginning, has been founded in 2008 by my former employer.
Matthias,
yes?
Just wait, are you changing your slides? Because I only see the first slide.
Only the first one? Okay,
yes.
Oh, then I have a problem with the presentation mode.
Do you want to reshare, like maybe just try to share again? Yeah, okay, this moved. Yeah, it moved.
Yeah, this is not the presentation mode. Maybe it's better, but then we are on the safe side. Can you see the slides? Can you read them?
Yes.
Yes.
Okay,
you can see the slides and read them. I think this is good. Thank you.
Yeah, then let's stay in this mode. Okay. So actually, I mentioned this, my former employer was Max Planck Innovation, which is the technology transfer organization from the whole Max Planck Society. Max Planck is huge. They have 80 different institutes in Germany, and about 40 of them are concentrating in life science. There's a lot of, let's say, project ideas, let's say, from different disciplines. And when I started in 2006, actually at Max Planck Innovation, there was no way to incubate these projects properly. That's why one of my projects was drafting a concept and executing it. And we have founded successfully the Lead Discovery Center in 2008. And as I mentioned, the first 10 years, an exclusive, let's say, incubator for Max Planck projects. But in 2019, we had our time change, so to say. We have actually generated a fund here within Europe, which is called the Kahn Technology Transfer Fund, #1 these days in 2019. And actually just recently, we have published a press release 2 weeks ago. We have been able to raise another fund. And in total, we have raised, let's say, 120 million euros, which is, let's say, on top added. So Max Planck is actually co-funding their own projects. This is really an exception, so to say, because Max Planck is quite a wealthy organization. They can do this. But these 120 million euros, we can really apply, let's say, to project proposals coming from academia. So this is the first important take-home message. We're not advertising this and want to have your money. So we have our own funds. We're really looking for high-quality projects and interesting new assets, let's say, to be incubated at our incubator. So we have an extremely broad academic network. Meanwhile, I will have also a slide later on for you. So in the LDC in a nutshell, 116 employees, the vast majority really PhD level and training from pharma and biotech. This is very important. So we are not an academic drug discovery center. We work on academic assets, but we have really collected the people from different, let's say, industry partners, from big pharma, medium-sized pharma, or biotech companies. We're always working on about 20 active drug discovery projects in different stages. We have a broad range of indications. We're really indication agnostic. At the end of the day, the medical need drives also our interest and, of course, the probability of success in commercialization. So some of the indications are not so attractive, but actually almost more than 90% of, let's say, what we are doing here. And we have a strong expertise in small molecules, in monoclonal antibodies and ADCs. So we are not working on cell therapy, not on gene therapy. So we have really a clear focus. I think this is very important for, let's say, a kind of a small incubator that we are. We have extremely flexible partnering models with industry investors and academic partners, and currently 3 clinical programs, let's say, with our licensees. And this is a kind of a schematic, I think, that describes what we are doing. So the house is the LDC. So this are the disciplines that we are doing. Normally on the academic partner side, there is the target identification side, the target validation side. But when it comes already, let's say, to the need for a library for high throughput screening, so assay development is being done jointly together. So with the academic partner, we have our own library in-house. I can tell later a bit more about this one. And once we have, let's say, identified really starting points for chemistry, which we call a hit compound out of this high throughput screening, of course, this is really the time when we invest heavily from our fund, so to say, because then we really need to integrate all these different disciplines. It really comes really to a budget. This is in the area of more than €1 million, let's say, per year that we are investing here. And turning these hit compounds into lead molecules. This is really the major, let's say, I think, success story that we have done in the past and that we are doing that is very challenging. A lead compound really means that it really shows a proof of, let's say, proof of concept in a relevant disease model. So at the end of the day, you really start talking about drug-like features. These are really compounds that can survive metabolism and reach, for example, tumors when we talk about oncology, and really cure, let's say mice, or even that can lead to tumor shrinkage. So once we have really generated these results in these preclinical models, then we talk about a lead compound. A lead compound also needs to be novel, which means we have this composition of meta potential for creating new IP. And also very important, we need to have a clear strategy for lead optimization. This is the last phase of medicinal chemistry, leads to what we call a candidate, a preclinical candidate. And this is the area also where we seek really to join forces with our future clients, customers, with pharma, biotech, or let's say investors. And this is just the academic network. As you see, in the beginning, we had only the 32 Max Planck Institutes on the top left corner of this slide. But meanwhile, we have about 180 academic contacts. We're not concentrating on Germany and Europe only. We have also at the moment really more than 24 universities already overseas. So therefore, I think this is also the idea of reaching out to you and to other universities in the States and Canada. And we also talk to Australian companies because we want to advertise this. And also we are also happy to collaborate, let's say, outside of Europe and Germany. Indications, this is just really statistics over the last 10 years. Still majority in oncology. Why? This is just simply due to the fact that we have access to a lot of oncology research going on. You get a lot of research funding, of course, also in your institute, I'm sure. That's why we do see a lot of, let's say, oncology assets. And also I think it's still very attractive for commercialization. So that's why we still have this, let's say, 40% of projects in the oncology area. But as you see, let's say in the rest of this pie, we're also working on neurosciences, also in, let's say, psychiatric diseases, a bit of infection, metabolism, fibrosis, and also cardiovascular diseases. We're not shying away because the incubator, what we are doing is applicable, let's say, to all kinds of indications. Whenever we need support regarding indications, we collaborate either with clinicians or with CROs having, let's say, certain proof of concept models in place. So anything that we need, let's say, to be indication specific, we are collaborating on, but the core infrastructure is applicable to all kinds of indications. Yes, and actually what you see here is actually a summary of all our success stories. In the middle, you see again the value chain in drug discovery, what we are doing, let's say, and as you see, we have these business models established that we do a lot of company creation. This is the lower box on this slide. We do this, let's say, in all kinds of stages. I will actually have some slides in the next couple of minutes describing this a bit more detail. On the top right box, we do the straight licensing deals. We identify big pharma companies, biotech companies. We do not have a territorial scope. We do a lot of deals in Asia. In the middle box, in the top middle box, you see, let's say, the collaboration partners. Very often also, let's say, venture capital partners or companies are asking us also that they can use our infrastructure for also devalidating some of their project ideas. We are also open for this kind of collaborations. And of course, we also do option-based deals, let's say in the beginning, when the hypothesis of the project is so attractive that companies want to secure kind of an exclusivity. So this is really what we have done over the last decade. You see, we are quite busy, let's say, in commercialization and have also, let's say, created a lot of, let's say, employment also in the startup companies over the last years. And what you see here is really, I mean, this is something we are always faced on, the feedback that you get, let's say, from different, let's say, potential clients, is that, of course, everything you offer is always too early. It's too early stage. You can go, let's say, into the clinic and you also might get the feedback that things are too early stage. But when you see, let's say, the potential clients that are structured in the lower part of this slide, let's say that you see that most of the partners really say when you have something in the hit to lead phase and you did not reach this lead nomination status, which really means proof of concept and relevant disease model, you're really too early stage. This is really nothing for serious partnering. So even when you are lead optimization, the bigger entities, the medium pharmas, the mega pharmas, they also would like to have, let's say, a kind of an IND ready package. But to all the others, you can really start talking on a serious level. So this is really something, I think this really is common sense. Anything before a lead compound, so to say, is really very hard to commercialize. And anything after the lead nomination, it gets more realistic, more serious. It's not never easy, of course, yeah, but the probability of success is significantly higher. And to understand what's going on regarding value generation, I have prepared this slide. So when you see, let's say, the black line, this is really jumping regarding the value level. So I think most of the academics generate a new target idea. But when you think about the value of this is really not very high. This is really very, very, very low. And even when you do really high investments in asset development, you screen a huge library, which is also comes with a lot of investment and costs, and you generate hit compounds. The increase in value is almost nothing. Yeah, this is very important. I think this is really what you have to realize, that the value generation is really coming in a step when you have, let's say, devalidated or validated some of these chemical series that you identify in the hit to lead phase. And when you come closer to preclinical models, you do your pharmacokinetics, your pharmacodynamics, and you prepare your proof of concept model, then the value is, let's say, jumping. And at the end of the day, as you have really a significant raise in the value when you have this lead nomination, but when you follow this green dotted line, this is really the investment. At the end of the day, you not break even when you have generated the lead compound, which means you need a higher investment and you do not really generate the value that you're dreaming about. So you have to continue with this project. We do lead optimization, and when you have, let's say, the so-called preclinical candidate, which is called here PCC, then I think this is the area where you're the first time break even. This is a value you can realize on the market, either, let's say, with company generation or straight licensing. So this is somehow, now you're coming, let's say, into the positive cash balance, so to say, on your balance sheet. So this is just really to face reality. It's really something that people are shying away. And this is the reason why industry and venture capital is not entering this area. That's why we have created an incubator and have also, let's say, collected a certain a special kind of investment, so to say, into this, which is associated with a higher risk structure, so to say, because all the expectations that purely financially driven investors might have in this phase will not turn out successful at the end of the day. So this is something we all have to realize. And that's why there is a kind of a gap. So people are talking about the valley of death, so to say, in drug discovery. This is the reason why this is the case. Nobody wants to invest something where it doesn't get the returns. And of course, you have also a high attrition rate. You will lose a lot of projects in between. Okay, so this is, I think, the business models that we have established and which I think is really realistic. So whenever you have, let's say, your lead compound nominated, which means this is the red box here, which is called licensing, you are in lead optimization, but you have a preclinical validation, then you can talk about also higher upfront payments, you can get about early stage cash returns. You have mature medium pharma, let's say, as potential clients that want to license this, maybe some early biotech companies. When you want to generate a company, you can really start earlier on. I think anything where you have a kind of a chemical hypothesis in place, I would say, which means you have your hit compounds validated, you know which kind of structures you want to follow in hit to lead phase. This is really you can seriously start talking to venture capital, maybe in the very early stages, really talk to company creators, maybe to VCs that have an incubator. This is also a kind of an attractive, let's say, potential partner for this project. Or if you come really closer to the lead nomination phase, also the classical, let's say, VC companies that provide seed capital. Of course, when you are happy, you can also do these option collaboration deals at any time. So sometimes then, of course, the risk is mostly, let's say, on your side, but you already might have some exclusivity fees. And of course, a clear, let's say, commercialization potential already. So this is something, of course, you can do at all stages at the end of the day. It depends really on the target of interest, the project of interest, whether you can raise an interest. But of course, this is not really, let's say, a commercialization event at the end of the day. And I think in the next couple of slides, I'm actually discussing the pros and cons, what you really need to commercialize successfully. And you will see this, let's say, in this one, I have on the left, let's say, column is really the topic I'm discussing. Then you have the three business models, these options, the license, the straight license, or let's say the creation of new companies. Regarding the unique selling position of your project, this is very simple and easy. You need to have either a first-in-class potential, FIC, or a best-in-class potential, but you need to differentiate. You cannot do something that other people are already doing. I mean, you really need to have a clear idea what differentiates your asset, let's say, otherwise you will not be able to raise any interest out there. This is very important. So, of course, it's ideal to start as a first-in-class, but sometimes there is already maybe a compound against the target of interest available, but you found the clue, so to say, to make it better, maybe a different kind of interference point in the same target, which comes with less adverse effects, whatever. Then I would call this a best-in-class strategy. This is, of course, very attractive when you can really compare it head-to-head to the first-in-class. Yes, so this is really something that is very important. I think if this is not the case, you can simply, let's say, forget and save your resources, investments for other projects. It's very hard to sell if you do not have, cannot tick one of these two boxes. But the target hypothesis. When you do an option collaboration, of course, it needs to be really strong because they are buying into this target hypothesis. This is very important. You might start with what we call a phenotypical screen, but you need to show that you really identify also a target. It's very hard these days to sell projects that just have, let's say, a cellular screening as a basis, and you need to do the target identification later on. So this is even stronger in a straight license or startup company, because people do not want to invest into risk-associated projects. And you might have very good compounds at the end of the day, but if you cannot really show how you want to optimize them, I think, and you need to apply technologies that work on the target. This is a very important point. You need to have a strong target hypothesis. By the IP position. So if you do a collaboration option, of course, this might or will be jointly generated. It's fine. You can also start just with an idea. At the end of the day, to execute the option, of course, you need to generate strong IP position. You need to have a freedom to operate. But when you talk about a straight license or a startup company, of course, then you need already to have this strong IP position. Investors invest into this asset only when they have kind of a guarantee that they have, let's say, a strong IP position. The same applies also in licensing discussions. People want to do due diligence, they want to see a patent application, want to ask a patent attorney about it. So therefore, that's why you need to have this very strong IP position. It's not so important in this collaboration type. Of course, you can generate it jointly. Indication swap. Why I'm saying this? Because many project ideas are coming with an indication, but in almost 1/3, up to 50% of our projects, we are swapping this because we do learn over time that this target might play a much better role, let's say, in other indication. And when you are talking to a, let's say, unflexible partner too early on, he might be really limited. When you think about an option collaboration, a company is interested in the target and the disease. But when you do see that there's another indication much better suited for the target, and then it's very hard to turn this around. And this is, of course, something that is extremely flexible when you talk about a startup company, because the investor at the end of the day wants to grab the full, let's say, financial potential of this project, regardless of the indication. So this is definitely something when you're not 100% sure, so to say, and you might explore not only one particular indication, then it's, of course, clearly in favor of creating a new company. Then you can really leverage the full potential in all indications of your target of interest. Financial terms, well, it's I think somehow goes without saying. Of course, the most attractive one is the license deal. Of course, you get attractive upfront payments. There is really cash return. That's why everybody wants to have, of course, licensing deals. All the investors, tech transfer, and so on, because of the cash returns. Regarding the startups, you have, of course, kind of what we call biodollars. You have shares. There's just, let's say, a theoretical value in it. But later on, you have, of course, an enormous exit potential once the company is successful and is becoming sold, let's say, to a big pharma company, for example. Then, of course, you can realize, let's say, in a one-time payment, enormous amounts of money at the end of the day. And for the option-based models, of course, goes normally with low entry fees. Risk is being shared, backloaded terms, and so on. The level of confidence, I think this is also something very important. When you do have a license, you normally have this so-called NIH syndrome, this not invented here syndrome. You're fighting against, let's say, internal opinions. It's very hard to convince the teams in big or medium-sized pharma companies by your project, and also keep them, let's say, excited about them. So you can also do a licensing deal, but maybe 1 year later, the project is under review again, and you might get it back. It happens really very often. So that's why I think this level of confidence in a straight licensing deal is not so high. It's much higher in startup companies because you have a highly motivated founders team. They have shares in the company. They fight for it. They do everything, let's say, to make this really a successful story. And this is really, the level of confidence is much bigger. In a kind of an option collaboration model, you generate this kind of project champion on the other side. This can be also, let's say, of a benefit. Level of control, maybe not so important, but when you license something straight to a pharma company, you have absolutely no control, so it's almost nothing. Of course, when you, let's say, as a licensor, so to say, as a university or tech transfer, when you have shares in a startup company, of course, you have, let's say, a vote. So there is a kind of a medium, let's say, level of control. Of course, in the option collaboration is very high. You are really normally a collaboration partner on highest level, and you can decide, let's say, jointly with your partner what you need to do. Well, this was really just my slides, let's say, regarding potential business models, and to demonstrate that this is really working. So this is actually the assets that we have. Also in my department, I'm heading the business development department. These are the assets where we're looking for partnerships. So these are the assets in commercialization today. Let's say for the Lead Discovery Center, you see on the left side on these arrows, you see our partners. Of course, the arrow means really the maturation grade of the project. You see when we have this dotted line, the lead level with this yellow plus, which means we have shown proof of concept, we are in lead optimization. So we are talking to companies and investors for all these projects, but we have not successfully commercialized them. So they are all available, so to say. And this is just to give you a flavor about the outcome of numerous academic collaborations where we are currently trying to identify partners. And so you see on the right side the indications. You see we have really a wide variety of indications and are absolutely indication agnostic later on, let's say, regarding the destiny of the project. So if there is this is really, let's say, the end of part one, so to say, regarding, let's say, the LDC as a setup, the business model, our success models, and let's say the different kind of business models. And when I'm coming now to the infrastructure, what have we really established? What are our 116 people are really doing? I think it's nicely seen here. So we have, of course, let's say a screening department. We have a medicinal chemistry department. I'm coming also with other slides into this in a minute. And what we do is really anything to preclinical candidate nomination. What we cannot do is really animal work. We have to do this with CROs. It works nicely. We don't do GLP tox studies, which means it's the regulatory preclinical study. And of course, we cannot do any clinical trials. We need partners for this. We have integrated all kind of state-of-the-art AI technologies, which is very important when you think about modeling and computational chemistry. So this is all state-of-the-art. And well, assay development, this is really something that where you can do really a lot of things really wrong. At the end of the day, you need to achieve medium throughput or highest throughput because you want to apply a huge number of potential small molecule compounds in a library into it. Ideally, you have these 1536 well-format plates, which means there are some requirements regarding the assay. We do biochemical assays, we do cellular assays, we do phenotypical assay where we do not have a clear defined target in the beginning. So this is something we have established. You can imagine that we have quite some experience. In these 16 years, we have started more than 120 drug discovery projects, and they all came with an assay in the beginning. So this is, we never had a dropout in assay development. This is very important. What's also very important is protein production. When you talk about a biochemical assay, it needs to be really reliable. It looks and sounds easy, yeah, but it's never easy. That's why it's very important to have also, let's say, the experience and how to create, let's say, a stable batch of your protein or target of interest. Our library is 260,000 compounds. It's really, we have collected from commercial vendors to increase, let's say, the diversity. Of course, we have collected, let's say, over 16 years, our own projects that are really novel compounds. These are really LDC project compounds. We have access to compounds from the Max Planck Society. We can use them also for others, and this is also a huge library. We're talking about bioactives. We took natural compounds. We have collaborations with, let's say, marine institutes in northern Norway. They have, of course, a library, let's say, from marine organisms. So that you see, we were very flexible in this area. When we have the feeling we need to increase the diversity, then we are collaborating, let's say, with commercial vendors and increase the library diversity. Well, then once we have generated these hit compounds and identified, let's say, and successfully screened, let's say, the assay of interest, I think then I would say the real work really starts, because then we have these hit compounds. The decision needs to be made which structure, let's say, to pursue, which one to drop. And here, this is really something you need really experience, let's say, from the team, let's say, to work on this. And MedChem is a never learning, is an ever learning, let's say, effort. So I think MedChem people that have just worked 5 years in industry have seen maybe 8 to 10 projects in their lifetime. That's a complete difference when you talk about really senior people that have worked 30, 40 years in MedChem and have seen almost everything, so to say. This is really extremely important to apply a lot of experience, because when you, let's say, walk with your compound in the wrong direction, it's very hard to make a U-turn and go back from the start. You will sink a lot of money into this. So this is a very important decision point, so to say, which are the chemical structures to really pursue, which one to explore and which one to drop. And of course, the hit compounds you see on the left part of this slide, they all start with a kind of a potency. They have been identified regarding potency on the target, but when we optimize them, when we make them a lead compound or later a drug, you will see what we have to integrate. We have to talk about safety, metabolic stability, solubility, of course, absorption, and potency is only one parameter. You do not have to lose the potency, of course, but you need to find a common ground that is acceptable for potential patients in the future. And this is actually your goal that you pursue, that you follow, so to say, in the medicinal chemistry phase. So turning it into a lead compound really means this is a multi-parameter optimization. You are monitoring and test your compounds in vitro potency and selectivity. We have to monitor the physicochemical properties, which means solubility, log-P, permeability, chemical stability. You have to monitor the biopharmaceutical properties. So regarding metabolic stability, protein binding, CYP inhibition, bioavailability, specificity, and so on. And you have to assess, of course, the modulation potential. At the end of the day, you want also to reach a composition of matter, novel patent fields, so to say. So as you see, there's a lot of disciplines a MedChem guy has to integrate when he suggests new analogs. And this is, of course, something that needs to go along with really a long experience, a long track record, so to say. This is hardly doable, so to say, with junior people, and almost not doable in an academic environment, at least to our, let's say, experience. Why do we have also biology? It's very important. We need to reproduce some data. We do some target revalidation. We have cellular pharmacology established. We have, let's say, we do the evaluation of efficacy and toxicity. We do mode of action studies. We might learn something over time about the target. We might need to have a better understanding. But this is, of course, let's say, a field where we are also closely collaborating with the academic providers of the project. So this is something that's where we are actively working with the biologists. Normally, let's say the projects come, let's say, from a biological background, but also biomarkers and target deconvolution is very important. We're doing this also in this department. And yeah, pharmacology. Very important, let's say, to assess the physicochemical and biopharmaceutical properties, safety, and so on. The so-called ATME, let's say, properties, which means for absorption, distribution, metabolism, and excretion. This is very important to monitor. We have all these assays, let's say, these are really standard in vitro assays. We have them running all week long, so to say, and our project managers can give compounds into this department. It's very important that you permanently deliver, let's say, these data points to the medchem people that are designing novel analogs. And then, of course, when it goes into the direction of preclinical testing, in vivo testing, very important to monitor the pharmacodynamics, which is actually the drug action and mechanism. So this really means what the drug does to the body. And on the other side, the pharmacokinetics, so to say, what the body does to the drug. It's about absorption, distribution, metabolism, excretion. All of these data, of course, have to be monitored. It's really a decision point, really when to do this, because it's not only time consuming, it's resource intensive, it's cost intensive. You can't do this with all your compounds. You really have to make a good selection. Otherwise, you're sinking a lot of capital. So that's very important. So the hallmarks of a lead compound, I think I can skip this. I was talking about this proof of efficacy in a relevant disease model. You need to achieve this novelty, let's say, regarding, let's say, the chemistry. And once we have nominated a lead compound, of course, the next phase is so-called lead optimization. We call this fine-tuning of the compound. We have already an idea how a drug should look like. We call this a target product profile. This is something you need to achieve when you nominate this for preclinical studies. And this is really, let's say, fine-tuning with the same parameters, the chemical structures that you have. It might take 12 months, it might take 24 months. It really depends on a bit also on luck, so to say, that you have made the right decisions in the hit-to-lead phase. So this is something where we normally start integrating also our potential client, pharma partners, biotech partners, and experts maybe from startup companies, because here it's very wise to already to apply a kind of a clinical perspective, what you would like to see in the clinic. So this is definitely driving the so-called target product profile a lot. That's very important to have at least clinicians, let's say, also involved into this phase. Besides small molecules, we also work on antibodies. We have an antibody discovery unit. We call it LDC Biologicals. So we do classical antibody discovery. We do antibody engineering. We do, of course, the IP protection, and we do the lead selection. We do the same with small molecules. Also, we optimize these antibodies until they have a clear effect in a preclinical model. And we do the same regarding commercialization. It's a bit different regarding attrition rates, and so good antibodies are a bit easier to sell compared to small molecules, because the attrition rate later on is definitely lower. Once you have a good antibody, you hardly lose it. It's different with small molecules. That's why we like to balance our project portfolio between antibodies and small molecules. Of course, it's all about, let's say, the targets at the end of the day. Ideal targets for antibodies are ligands, receptors, ion channels, and GPCRs. So, but we also, let's say, work on nanobodies. So in the future, maybe also we are targeting more cellular targets. We do monitor some technologies already there that might be doable in the next couple of years. But at the moment, we have a clear focus where we do have intracellular targets. We work on small molecules. Where we have receptors or ligands, we work on antibodies, more or less. Of course, there is a combination of both. These are the ADCs, and we have both expertises established. So therefore, we are, let's say, doing also, we are linking, so to say, also payloads to antibodies. And what's very important to do a proper, let's say, drug discovery process is that you have established two departments. We call it project management and also business development. So project managers, I would call them, they are the conductors, so to say, of the whole phase. They are, let's say, integrating all these disciplines. They are organizing it. They take care for the budget. They are planning, so to say, the steps sequentially that need to be done sequentially. So this is really something that is very important, that they have, let's say, an overview on all these projects and not let's say, these are not really bench people. And what's also important, business development, that you do a constant competition watch, you do a market search and analysis, because what we are talking about from assay development to commercialization, we talk about 3 to 4 years. And if your target is very attractive, many things will be published. Meanwhile, during this incubation phase, competitors might arise. You need to do head-to-head studies. You need to monitor these kind of compounds and so on. And you need to constantly talk to your network, because otherwise you are optimizing a compound that nobody wants to have at the end of the day. Yeah, so this is the part, so to say, regarding the drug discovery unit that we have, the pitfalls and how we organize this. And yeah, what are we looking for? So how to become an academic partner? I think the most important thing is, it's just a small line on this slide. It's in the bottom right side. There's definitely no IP needed. We just need to have an idea. But this idea needs to be novel. So as I mentioned in the beginning, this unique selling position needs to be there. So there needs to be a differentiation point compared to other activities on this target or in the indication. So this is very important. I think this is the most important thing. So if the indication is attractive, the USP is there, and then we look for willingness to collaborate. Yeah. So what kind of systems are established? What kind of research tools are there? What can be done? Is there a mouse model established, a proof of concept model? This is definitely a big benefit, because it takes a long time to set it up if you don't have this. But that's it, so to say. That's why we're really motivating our academic network to send us really project ideas. We have a department, we call it our scouting department. We are looking for this. We come back with Q&A and with a questionnaire, so to say. And yeah, this is something most important is really nobody should really shy away telling us their project ideas. Of course, in the first instance on a non-confidential level, this is all fine. If there are publications out, it's all fine. At the end of the day, we need to sense whether there is really something unique expertise of the PI, so to say. Ideally, let's say one of the leaders in the field, so to say, also, let's say, knowing exactly what's going on in other academic labs. So this is more or less what we are looking at. And at the end of the day, regarding commercialization, we are sharing the proceeds. As I mentioned, we do not need investments. Of course, we try to jointly generate some non-dilutive fundings, check whether there are some public grants available for this project. This is of course clear, but if this is not the case, we like the project, we have the capital to incubate it. And at the end of the day, we are sharing the proceeds. This is something that we are negotiating in the beginning, normally with a tech transfer unit. And as you see, we do this 16 years now. So we have always agreed in numerous universities and institutions, so to say, on a fair share at the end of the day. Yeah, that's more or less, let's say, what I have to tell you. And I stop sharing my slides, and I'm happy to take your questions.
Nice, thank you. That was very informative. Are there any questions from anyone? The person from Biocytogen or any students? All right, so while I think people are thinking of questions, Matthias, so I want to understand a few things. One is that the slides were very informative and very well organized. So you mentioned that there were several programs that were where you were looking for partners, and they were in all different disease areas. So are there specific modalities and specific disease areas where you see repeat success in your hands or in your at LDC?
Yes, definitely, yes. So as you've seen from my indication pie, oncology is, of course, still very attractive. This is just, yeah, well, there's a lot of oncology research going on. There's still a medical need. It's getting more and more complicated in oncology. I think there is really The access to patients is really becoming more and more difficult, but this is not something that should really drive our strategy, so to say. This might be a solution in the future for this. But oncology, of course, regarding the therapeutic window you need to achieve, we have still a lot of flexibility. So in oncology, when you have really a patient population that is really almost untreated, you need to show efficacy. And actually, when you have manageable adverse effects, that's doable. When you think about a pain drug, pain really means you have no adverse effects. You just need to address the pain at the end of the day. So this is the hurdle for commercialization is much higher. So it's also in psychiatric diseases. It's first of all, the predictability of the animal models is almost not given, which means you are doing something, but at the end of the day, you have no idea how this translates into patients, into humans. So that's very hard to commercialize. You have to go all down the way, maybe via creation of a startup company to show clinical success. In oncology, this is really different. The same also in cardiovascular diseases, where you have an immediate, let's say, effect, and it's all about the predictability of the animal models you do. So if they are really giving you an almost safe prediction, what you can expect in humans, that's of course something of interest for us.
Okay, that helps. So in terms of, so I see that you guys are looking for different types of projects. So when an academic is thinking about approaching you, what should they present as a package? Like, what should they come to you?
Actually, a one-pager description, what they're doing, especially regarding the mechanism of action that they want to interfere, about the targets, relevant publications, that helps us, let's say, to get into the story, so to say. I think, according to our experience, we will identify the pros and cons of this project very soon. We have an idea about potential targets, whether they are druggable or not. We think about the modality, the best of modality, and so on. If there are research tools already being tested and there is some preclinical support, knockout data, very helpful, of course. If there is a genetic linkage, this is always very good. If you identify for a rare disease, let's say you have a genetic linkage in your target, of course, this is always of interest. Yeah, I think the more information you can provide describing the mechanism of action you want to tackle, this is ideal for us.
Okay, and if so we do have a few labs. So do you also look at drug repurposing projects?
Oh, we don't like them, to be honest. Yeah, so we never did them, and we are shying away from this. I know that many people do this. It looks like a fast success, so to say. It normally never is. Yeah? So therefore, because it's a question, it's a very nice research tool. It's a good validation tool, but there are so many, let's say, parameters. We all know only a few success stories of this, but many ideas of doing that. So we are shying away from this. We're normally not doing this.
Okay. So that was very clear and precise what you explained. So then the next question is that I understand with say the way you explained, say access to cancer patients versus coming up with these endpoints for pain or other things. So that said, what if say, so we do have medicinal chemists across the university. And as you know, they love coming up with new chemistries and stuff, right? So how do you engage with those projects, or you just want to know about the mechanism and you don't really worry about? So is there any value in a medchem lab coming to you with what they have versus say somebody just coming to you with the molecular pathway they want to target?
Yeah, well, actually, of course, we're talking also to a lot of chemistry-based institutes. They generate new compounds, most of the time against validated targets. So it's not the department that discovers novel targets, of course. We are looking at this. There are some hurdles, of course. Most of the time, the people do not want to disclose the chemical structures. And then the question comes, what can you evaluate? Yeah? So we are shying away entering into CDAs and seeing structures from others. It's a kind of contamination. We have so many projects we are working on. There might be an overlap. That's why it's not a good entry point for us. Yeah? And but if you can show a nice data set with these compounds that are convincing that you really can differentiate from maybe other drugs that are directed against this target, that's all fine. We have a look on this. Yes. It's a bit more complicated regarding disclosures normally, yeah, because then the question really is, what are you medchem people really requiring for doing the next steps? Can they do their own, let's say, preclinical studies? They can also, let's say, give an order to a CRO for testing these molecules. But the question would then be, what is really required? Because the LDC as an incubator has been set up definitely for, let's say, biologically orientated researchers that identify really novel targets. And we have a medchem department established, but if there are institutions that have also a similar setup, the question really is, how can we contribute, so to say, to the next step of incubation? So this is really something we need to figure out, depending on the status of this medchem project. Is it really close already to lead nomination? Then the question is, why are they needing us? Yeah, or is it just maybe that you generate some hit matter against the compound, and still the majority of the medchem phase is lying ahead of us? It really depends. So we're looking on everything, but just to manage the expectations, it's a bit more problematic normally when we do see novel chemistry.
Okay, all right. So the other question I had was about that slide where you showed different options that you engage with. So like the option or the collaboration, the licensing side, and then the startup. You also mentioned that LDC is now doing more of startup creation. Can you talk about that? Like what is driving each step, or like why do you focus on one versus the other more?
Yeah, I think this is really a result of this series of slides where I have shown the pros and cons. So what we do see is that in these days, pharma companies are looking for more and more later stage projects. Yeah, maybe it goes with the general economic we are facing. We have a lot of inflation and biotech companies suffering getting capital. They need to sell their assets. So a lot of good assets are being offered to pharma from the biotech scene globally, which means, which results in that they are really saturated with project ideas and not looking for early stage ideas coming normally from us. So that's why we think that's a quite volatile, let's say, phase we're in at the moment. It's easier for us creating new companies compared to straight licensing deals. 5 years ago, it was completely different. Yeah. So at the end of the day, I have to tell you that licensing is definitely preferred on our side. It's much less work. Yeah. For startups, you always need a team. I think it's hard to create a company without a team. Normally, the PIs do not want to enter these companies. You have to find people. They are rare. Yeah, they are expensive. So it's not so easy. It's not like you go left or right. Yeah, so it's not that easy. That's why you have really to check, do you have a team available for this project? Does it make sense? Can investors really trust this? And so on. So that's why it's, of course, there is a lot of complexity in it, and creating new companies comes always with, I would say, 10 times the effort compared to a straight licensing deal.
Okay, that was very informative. So in terms of, say, like allocating resources, both time and funding from your pot, so how much do you put in, like how many years, how much funding before you make like a decision on whether to keep continuing versus folding the program?
Yeah, I think really from let's say the start of a project until lead nomination, 2.5 years. Yeah. And we say something like close to 3 million euros, which is at the moment like dollars. And for lead optimization, additional 1.5 to 2 million euros. So it can be really up to 5 million for the whole phase, which is really 4.5, 5 years. And yeah, so this is what we are investing into these projects. of course, milestone dependent. Yeah, not all of them get the full investment. Some of them drop out, and some of them are being commercialized early on. This is also, of course, something that is attractive when we create companies. This is really the attractive side, that we get an additional investment, so to say, from other investors, that at the end of the day, we are offering then the LDC infrastructure as an infrastructure also for these startups. And but we create, let's say, higher volumes of investment for these kind of projects then.
Yeah, all right. So final question, like, do you have like an ideal asset or an ideal kind of modality that you would like us to search for across Northeastern?
I think antibodies and small molecules, I think antibodies is a bit preferred, yeah, because as I mentioned, it's maybe hard, let's say, to identify one, but when you have a very good antibody that shows efficacy in a preclinical model, you will hardly lose it later on. So attrition rate is very low later on, which means you can create more value, let's say, in any kind of commercialization. And so therefore, but they are hard to find. I tell you, when we are scouting, so 9 out of 10 project proposals is relying on small molecules. So that's why I would say we always look for new antibodies. That's fine. But you will not, it's not a 50-50, let's say, balance what you see in academia.
Okay, all right. Well, thank you for your time. And I think this was very informative. Will you share your slide deck with us and we can circulate it?
I can send it to you. I will do this.
All right. Thank you.
Perfect.
Thank you.
Thank you
Nice
so
meeting
much.
you. Bye.
Thank you. Bye-bye.